Thank you for the introduction. I hope not the last, but the latest. And today I want to speak about endurance. Um, when we think how the brain encodes the world around us, one of the most prominent idea is the idea of the neuronal selectivity. To be responsive to the whole bunch of what organism can detect around. Instead, um, mhm. Instead of this, if we have a look on each particular neuron, it will encode some distinct feature of the environment. And historically, the states that the individual neurons is not only the structural basis of the brain, but also the functional unit of the brain. And this led to the idea of the selective neurons and its receptive field. Receptive field is some some view of the feature that is most responsive for these neurons. For that, the neuron is most responsive to. And the pioneers of the selective neurons idea was David Hubel and Thorsten Wiesel, who in more than 60 years ago discovered the selective neurons in the cat visual cortex. And this was the neurons um, that were specifically tuned to the uh, tribes of different orientation. So that the selective neurons have been found across the brain region, each tuned to the distant features of environment or behavior. And the top of this selectivity can be described as the concept cells that were introduced by Quiroga back in the beginning of the century. And the example of such concept cells are the cells of Price, Jennifer Aniston cells, and so on. And maybe a lot of you have seen this video many times. So I want to show it again. So the recording from the human brain, and due to the medical reasons, the patients needed to undergo this surgery, and the electrodes were implanted to the brain, and not only some medical issues were resolved, but also the researchers can make the direct registration of individual neurons when they showed patients some pictures or some videos and so on. Maybe you can turn it on. And now we can see the firing of a single neuron from the internal cortex while this person looked at these video fragments. So you can name, so this neuron is the Simpson neuron. And you can try to imagine what part will be here, what video fragment. Okay, there was a Simpson movie again, so and neurons were activated very quickly again. There is the other part, last part of this video where the researcher asked, what do you see? And he starts to remember, I saw the Sex and the City, no reaction of the neurons. Ah, it is here. I thought I have the cut version, sorry. So the Hollywood style, not the bigger. And now it remembers about the Simpsons, and the inference is activated again. So from all of this, we can claim that the selective neurons contain building blocks of neuronal encoding in the brain. So, and or not for, but into the discovery of the selective neurons in Union, provided the idea of the functional systems. And instead of describing the brain as the collection of individual neurons, he introduced the idea that there is a functional system that are based on the achievement of a result. And the result is achieved through the different behavioral acts. So behavior is produced by these functional systems that are dynamic, goal-oriented, and distributed across the brain and across the whole organism ensembles. System theory was the work made by the students of the Petr Kuzmich. So this was the idea that was developed by Vyacheslav Borisovich Shverkov, who introduced the concept of the specialized neurons. So these neurons become active in specific behavioral acts. And if we're trying to distinguish between these selective neurons and specialized neurons, we can say that selective neurons are defined as cells that respond selectively to specific stimuli or to specific class of environment stimuli. So this is the reactive paradigm. So there is some stimuli and there is active reaction of the selective neurons. And the specialized neurons are distinct from the selective, and they participate in distributed systems that integrate together sensory, motivation, and motor and other components to produce the organized behavioral that result in the activity of these neurons results in some adaptive for the organism reaction. So these neurons do not encode the discrete stimulus, some discrete features, but are involved in the systems that support integrated behavioral patterns. So this is the example of such specialized neurons from the lab of the from the Shverkov lab. Which is now the lab named after Shverkov, now is in the Institute of the Psychology. So they made their experiments in different species, also in rabbits, and they performed the electrophysiological registration of the activity of individual neurons when animals were doing specific behavioral, and this behavioral was to get some reward. It was the appetitive reward, and the animal needs to push on this plate or to pull the ring, and the reward was given to the animal. And so here you can see the specialized neurons that are active in every single behavioral act of the pulling the pedal or of the pulling the string. So this is the neuron of the left side, and this is the neuron of the right plate on which animal can push and get the reward. So this is the idea of the specialized neurons that are defined as the neurons of the behavioral acts. And we see we can distinguish selective and specialized neurons from each other. But of course, we are talking about the same phenomenon. All of these neurons are the same in the same brains, but we are describing them in a different manner. So now we can try to rethink these definitions of selective neurons and specialized neurons and found the truth of what these neurons are, what they mean for the behavioral, what they mean for the organism. And now I want to zoom in a little bit from the whole brain to the particular brain region. Oh, sorry, not yet. So, and when we're trying to think about these neurons like together, what they have in common, still knowing the differences between them, we can try to have some classification of the neurons, of such neurons, and try to find what types of selective or specialized neurons we can see in the brain. Can find the physiological selectivities or physiological specializations. So these neurons space, sensory stimuli, and so on in an experience-dependent manner during early development. And this type of the selective neurons include place cells, grid cells, the neurons with the visual receptive fields, and so on. Then we can find the neurons that are selective for specific behavioral acts, but independent of the outcome. So anytime when animal or human make some behavioral act that is the same, this neuron will be active. So this is like jumping cells, rearing cells, freezing cells, and so on. This is the example of the jumping cells that is selective for the jumping behavioral in rats that were showed by the Buzhaki lab, and this is the paper. Then we can find the neurons that are this is apparatus. So this means that it doesn't matter how you made the particular behavioral act, but what matters is the outcome and the possible reinforcement. So the examples of this include like cells of the ring, as I showed you in the rabbit, lever cells, again in the rabbit, and so on. And then they can be concept relative selectivities. And these neurons are selective for a concept that emerges at the intersection of the multiple distinct outcomes. And as an example of such neurons, we can name again the classical concept cells like Jedi neurons, Luke Skywalker neurons, and so on. And also like a nest material cells in a pregnant female mouse. So the pregnant mouse can make a nest from different materials or in different objects like paper or cotton, or to make a nest in some cup. And there will be a neuron of the idea of the nest. So if this object or this place or this construction is okay to be a nest for this female and her pups, there will be a neuron that encode any of these material objects and so on. So this is the idea of the concentrated cells. And now I want to zoom in from the whole brain to the particular brain structure and to talk more about the hippocampus. After the visual selective cells that were discovered in the visual cortex, the place cells that are hippocampal neurons, I think are the most known and well-described selective cells in the brain. So the place cells are hippocampal neurons that fire selectively when animal travels through or occupies some particular location in the environment. And each neuron has one or more place field. And the placed field is the particular place of the environment where this particular neuron has the big amount of time. And across a population of neurons that this field tile a space together to provide an internal map of the environment for free navigation, special memory, and so on. So the place coding is stable across days, but yet it is flexible. So representations, the particular place neurons are remapping, so they change their place fields, and also they can stop being or the place cells, and they can drift and change their place field across the days. These place cells performs the hippocampal spatial representation, and these cells allow animals and humans to explore the environment and to remember the environment. And it was well established that the majority of hippocampal selective neurons are the place cells, but to which extent is the majority is not very well agreed, because here you can see the graph from the recent paper from the Yaniv-Ziv group, and in this on this graph you can see the fraction of the place cells from the whole population of the hippocampal CA1 active cells, and you can see from very low amount here to very, very big amounts here. You can see here some rules about it. So usually if you perform the electrophysiological registration, the amount of the relatively new registration based on the calcium fluorescent activity, the amount of the place cells is less, but still the diversity of the fraction of place cells in different papers are very big. So, and Ziv and his colleagues in this paper, they argued that the all place cells, all active neurons of the hippocampal CR1 are place cells, and only there is no different populations like these are place cells, these are not place cells. All of them, according to the Ziv view, are the place cells, but some of them can encode also something else. And this leads us to the question what can be focused in the hippocampus. From one edge, say that all cells are place cells, but still we have a lot of examples when the hippocampal neurons encode very different features of the environment. So there are other cells. These cells encode specifically different odors that are presented in different parts of the environment, but not the planes. The jumping cells that I already showed you, the nest cells that were described in the 2017 time cells. So these cells encode the line of events instead of the line of the particular parts on the environment. There are also auditory and visual cells in the hippocampus that specifically respond to different auditory and visual stimuli. The cells that specifically encode the novel object cells, novel object in the environment, and the cells that encode social partners, like known, familiar, or a novel mouse, speed cells, and so on. This is, of course, not the full list. So, to some extent, we can say that the most known and well-established selective cells in the hippocampus are place cells, but still the hippocampus is full of the different other selectivities, like shown here. And now I will switch to our data, and I will show you our work about the hippocampal neuronal selectivity. So what can be encoded and how it is encoded. And I will start for the methodology for identification of the selective neurons. And then we'll show you two examples of the different behavioral tasks where we registered the activity of the hippocampal neurons and trying to find to what features, to what behaviors they can be selective. So the methodology starts from the automatic recognition of the animal behavioral, because the animal are doing different types of behavioral in different types of tasks in the environment. Some of them are not related to parts of environment, some are. So we need to track the behavioral in the animals in different behavioral tasks, and to be able to recognize the distinct behavioral acts specifically. And for this purpose, we use the model that is deep lab cut. This is a model that is based of the deep learning and the artificial neuronal networks. And what we need to do here, we need to once mark the target body parts. So this is the nose, this is the tail base, this is the limbs of the mouse. And then we can train the model, and after that the model will recognize the same body parts in other environments, in other mice. So we can once perform this marking of the target body parts, and then we will achieve all animals in all environments, in all behavioral tasks will be the body parts of these animals will be automatically recognized. So after that, we can perform the automatic recognition of behavioral acts. This is done based on the how the body parts changes its location from each other, and when we achieve the body parts detection by deep lab cut, we perform the analysis of the angular and radial velocity vectors of these body parts from each other. And this allows us to recognize and to automatically define different types of behavior. And here you can see the rarings. So this is the behavior when the animal have the vertical behavioral activity and starts to look around the environment to the walls and so on, like here. And this can be done by the relative position estimation of the hind limbs and the tail base. And here you can include in this automatical recognition the parts of the environment. This example is the open field. So this is the square arena with the 4 objects inside it. And these are specifically the moments of mouse interaction with different objects in this arena. So we identified the position of the nose here. This is blue nose relative to the specific object. And this is the Sphinx pipeline that was done by the Victor Plusnin, who is not here now, but will come to the OLGA Neuroscience Conference. So then it comes the time of the registration of the brain activity. And for this purpose, we use the calcium imaging. So the calcium is a correlate of the electrophysiological activity of the cells. And we have the fluorescent calcium sensors, and this sensor just binds to the calcium and starts to fluorescent the photons, the green photons starts to come out of the sensor, and we can detect it. And for the detection, so these are the cells with the calcium sensor. We implant here a lens. This is a lens just above the hippocampus. And this is the Miniscope. It is a small device. You can see it here. It is rather small compared to the animal head. And this is the small device with the CMOS sensor, with the filters and objectives, and it can record the video of the brain activity. So here are the bright circles are the cells, and the animal can perform any free behavioral, like you can see here. This is the mice with the implanted Miniscope, and this is the activation of its CA1 during the pre-exploration of the open field. After that, we need to define these cells in the video of the cell activation, and we use the Kaiman protocol and the Birmine pipeline that was made by Vovosatsko. And we can extract here the rows of the different hippocampal cells that were active during the whatever animal was doing. And here are the examples of the calcium traces. So each increase in the calcium activity means that the calcium at this time leaves the cellular depot and was in the cytoplasm of the neurons, and the calcium sensor was able to bind to the calcium and to become the visible bright and fluorescence. So here you can see that there are some neurons, some time when there was a small activation and some time when there was a significant increase in the calcium. So yes, this is not the direct measure of the electrical activity of the neurons, but still this is a correlate, and each calcium event here, each big increase in the calcium usually summarize some number of the distinct neuronal spike, like electric spikes. And so we perform the surgeries to inject the virus that carries the fluorescent calcium sensor, to implant the lens, and then to fix the base plant for the Miniscope. And during the behavioral experiment, we just attach this Miniscope, perform the behavior, behavioral task, then deattach the miniscope, and the animal can live its life with the only green lens and the small base plant. And so we can perform these experiments like to the end of the life of the animals, like for 1 year and 1 year and a half. And usually we make a surgery for some number of animals, like 20 or 30, and then each animal goes through the different behavioral tasks. And we can record activity of the merely the same neuronal population in different behavioral tasks. And then everything goes to analysis and to finding the selective cells. And for this purpose, Nikita Paspelov, who is here, developed the intense pipeline. And this idea of the intense is to calculate the mutual information between the two sources, one of which is calcium data, and the other is extracted different behavioral variables. And after we calculate this information, this is examples of the variables. So this is the head direction of the animals. And this is the events of the interaction with the objects. And this is the X and Y coordinates. So this variable encodes the position of the animal in the environment. And this is the examples of 3 different neurons and 3 different calcium traces. So the intense combines these 2 sources of data together, and then we calculate the mutual information, and we compare it to the shuffled data when the calcium data and behavioral data do not match each other. And then we select the neuron behavioral parts that are here showed in green that has enough mutual information, and they exceed our established p-value. So, and the result is the the result is these selective neurons. So this is the neuron that was selected for the freezing behavior. And here you can see the calcium signal in red, which was during the freezing behavior. And in blue, this is the calcium signal, which was at rest of the behavior, not freezing. And these neurons are selective to the center of the open field. So it was firing more when the animal were in the center of the open field, but not near the walls. So this again are another example. This neuron is selective to the object number 4. And these green lines shows us the events when the animal was interacting with the neurons number 4, and blue line is the calcium trace. And here again is the density of the calcium activity when the animal was interacted with the neuron object number 4 and was not interacted. Here I put this citation from the Shvirkov because this explains why in the beginning of the talk, I spent some time to describe the idea of the specialized neurons, but now I talk about the selective neurons, because this is because of the very strict definition that Shvirkov made for the specialized neurons, and some inconsistent or sporadic activation during a behavioral act are, are not sufficient for classifying a neuron as a specialized for that act. But here you can clearly see that, yes, these neurons, due to the mutual information calculation, was identified as a selective neuron, but still it has some activations without this specific behavioral act, like interacted with the object. And vice versa. Here it was the event of the object interaction, but the activation of these neurons was not so high. So this is why I will. This is why I call this. We call these neurons like selective, that are not specialized in the very strict definition made by Shverkov. So the neurons can have more than one selectivities, but the majority of them has selectivity only for one feature. But about 5% of all the neurons are multi-selective. From these 5%, most of the neurons that are multi-selective are selective for two different features. This is the example of such multi-selective neurons. These neurons was identified as a locomotion selective neurons. Locomotion are the movements with a high speed. And the same neurons can be identified as a selective to the specific head direction. So this neuron is activated when the animal is running somewhere with a fast speed, and also for this specific head direction. So now this is the second part. And based on this methodology I just described, I will show you our results on the selectivity of CA1 neurons in a very simple task. This is the task of the open field exploration with a decline of novelty. So this is like 4 days of the exploration of the open field. You already seen the open field. This is the square one, like 50 centimeters to 50 centimeters, and with 4 objects inside. So we made a surgery, then we have several weeks for animals to recover. Then we performed the handling. So the animals were attached to the Miniscope. And then we performed the registration during the 4 subsequent days of the novel open field exploration. Here are the behavioral results. So this is the distance traveled in the open field on each day. So on first day, they explored it more actively, and then you can see the significant decrease of the interest and of the exploration of the open field. So they start to spend more time in the corners, like here, and to spend less time near the walls. So we just exclude the corners from the walls. They start to sit more in the corners because they already explored this open field very well, and the novelty is declined. You also can see the decrease in the running events. Running is the movements with the high speed decrease in the object interaction. So the first day, the object was very interesting. They explored it, and then the interaction goes down, and some increase in the duracity of the freezing here. And here are the types of the different neuronal selectivity that we can find. So this is the distribution in 13 mice, and you can see that the majority of the selective cells of the CA1 in the hippocampus were the place cells. A big amount of the selective cells were the cells selective to objects inside the open field. And this is not due to the place. We checked this. This is the cells that are selective to the object, not the place. So also velocity, head direction, body direction, sometime near the walls, in the corners, in the center, some cells that were selective to stops, walk and runs, rare, freezing, and so on. Some cells can be selective a lot to the place, a bit for the body direction, and other information is undefined yet. So it didn't get in these features that we predefined for the study. Some cells, this example, the cell is selective to runs, a little less to walls, and some extent is also undefined. And now we can look on the number of the selective cells in different days of the open field exploration with the novelty decrease. Sorry, this is the examples of the cell activity during the different behavioral. Can you please start both of the video? Thank you. So this is the cell that is selective to the object interaction. And this is the cell that is selective to the space. So this is the place cell. And now we can see how the number of the selective cells changed during the novelty decline in different sessions of the open field exploration. So overall number of selective cells didn't change significantly, but we can see here that the number of place cells were declined. And this is almost the only change in the neuronal selectivity in the hippocampus in this task. So this encoding of the different movement types were stable. The same for the neurons selective to the different parts of the arena. The same, surprisingly, to the object selective cells. Still, we saw in the behavioral results that the mice tend to explore the objects less during the novelty decline, but still the was the same. There was some increase in the speed, responsive speed selective cells, and some increase in the body direction selective cells. So to summarize this, here we established the whole methodology to find the specific behavioral, to describe it, to find the selective cells, and show that the place cells in the CA1 were the most prominent selectivity type, and the second selectivity type was the selectivity to the object, and the proportion of the place cells declined across the days with the decline of the novelty. So it can suggest that there was a reduced demand for dense spatial encoding because the environment becomes familiar, and there was a shift toward more stable spatial representation. So only the necessary hub of the place cells remains active, and all other cells were gone. And also we showed that different types of selectivity were distributed across the population of the CA1 neurons and maintained stable at stable proportions over time. And this is the last part of the talk. And here you can, I will show you the our work on the CA1 neuronal selectivity for a familiar object. And this work started when we, with Ksenia, was feeding mice with the cottage cheese work with the cottage cheese, and we saw that they can use these cups that we give them for the cottage cheese in a different ways. They can eat from them, they can put their pups, they can sleep there. So we just put in our minds that animals can use these cups in a different manner, and maybe there are some neurons of these cups. So, and some years later, we started this experiment with the bowl task. It also was inspired by this work made by the in the Chen laboratory. So they showed that there are the nest neurons in the mouse hippocampus. Can you please also start the video? So these neurons were selectively firing when the animal was in the nest, and that can be any object in any place, but still the animal should recognize it and should think about it as a nest. So this is the same cup, but in a different environment. And when the animal is in this cup, the neurons keep firing. And here you can see the activity of the same cell in different types of cups, but all of these cups were recognized by these mice as a nest. And also there was a later work done with the different methods without the neuronal recording, but still here they showed that pregnant females, hippocampal CA1 of the pregnant females is activated based on the CFOS expression when this female mouse produced the nesting compared to some control object that was not recognized by this mouse as a nest. So altogether, this inspired us to try to find the ball neurons in the mouse hippocampus. And the idea of the experiments there was that the experimental group of the animals we placed in their home cages this bowl. For this time, it was without any food. It was the empty bowl in their home cages, and they can use it in any way they wanted. They can sit, they can sleep, they can put some food there, and so on. And the control group was without a bowl in a home cage. So they just live a normal life in a standard home cage. And then we performed this series of the exploration of the different open fields with a ball alone, like here, with a ball and a new different ball, with a ball and new Lego object, ball and inverted ball, like placed on the other side, and again ball and a new object in other environment. And we performed this automatical behavioral analysis. And here we can see the animal, and these lines means then when the ball is between these lines, the ball is in view of this animal. And can you please these two also start? And here we can see the animals inside this bowl. They like it and they tend to sit in this bowl in this open field. And this is the animal interacting with this bowl. He's not sitting inside, but instead can sniff it, whisk it, and so on. And this is the traces of the behavior. So this is the behavioral again around the ball, and this is the direct comes when the animals see the ball from here and start to run towards the ball very fast. And usually it after that it sits inside the ball. So this is the different type of the ball directed behavior. And чуть-чуть назад. Thank you. And you see that animals that were familiar with the home cages, they spend a lot more time with the ball during the different sessions in the open field environment. This is these pink groups compared to the green group. And they spent the first sessions more time during ball exploration, and they performed more direct runs, as I showed you on the previous slide here, more such direct runs to this ball compared to the control animal group here. And then when we analyzed the neuronal selectivity, this is here, we again showed that the majority of the cells are place cells in both groups. Also, there were a lot of velocity selected cells. And you can see that there were cells selective to the balls. And the distribution shows that there were more such cells in the ball group compared to the control group. And here you can see the dynamics of the number of the selective neurons for different behavioral acts. This is neurons that were selective to the sitting inside the ball. This is here. And there were more such neurons in the ball group compared to the control group. And these are neurons that were selective during the for the interaction with the ball. So these neurons were active when the animals sniff or whisk the ball, or look at this when it was nearby the ball. And again, there are more such neurons in the ball group compared to the control group. And here are the features of the selectivity that are not ball related. These are two examples, like place cells and locomotion cells. And you can see there is no significant difference between the number of such selective neurons in the control and ball group, nor in neurons that were selective to the specific locations that were place cells, nor in the locomotion selective cells. And again, I just didn't show it here. Any other no non-ball selective neurons, the number of such neurons were not differ between these two groups of the animal. So only there was a bigger number of the ball-related selective neurons in ball group compared to the control group, which means that there was some representation of this ball that were established during the home cage, during the time when the ball was in the home cage. And the last result from here is the number of the ball selective neurons that was the ball selective for all 5 sessions. So this is rather stable ball neurons. And there was 5 such neurons in the ball group of minds. This is shown here. So these are the 5 neurons that keeps their selectivity for all the 5 sessions. There were 17 neurons that were selective to the ball in in 4 of these 5 sessions, 52 in any 3 of these sessions, and so on. And still in regardless of number of sessions where neuron was ball selective, the number of such cells were higher in the ball group comparing to the control group. And this is the same for the fraction percent of the cells from all active cells in these 2 groups of animals. So together, we show that animals that are highly familiar with the ball tend to prefer to sit inside the same ball or to remain nearby to the same ball in the open field in a new environment. This behavioral is also accompanied by an increased number of CA1 neurons that are selective to the ball, and other types of neuronal selectivity remain unaffected by whether the animal had long-term exposure to the ball or not. This means that these life in the home cage with the ball just take more neurons from the CA1 population for the ball selectivity, but it does not affect the other types of the selectivity, and these codes remain the same. So this is this is the end. This is what I wanted to show you today in this talk. So this is some possible questions for discussion. I will show it later. So this is a big thanks for the members of the lab who performed all this work together with me. And this is the last slide. Maybe you're interested to work on coding and analysis of the behavior or neuronal activity of some mouse work. You can come and ask me or Ksenia, Anya, or Nikita to talk about some possible work together. So thank you.